There we go. So starting through this, we're primarily going to be looking at diffusion and like what's involved with that. Again, there's a whole lot of keywords here, right? Hypotonic, hypertonic, isotonic. What the heck does all this mean? So quite a bit. Cells is probably the most complicated unit we're doing. So like, just so you guys know, after this is done, like you've climbed uphill. It's downhill from there. So basically, so whenever we're thinking about like particles, they're always in motion. When you boil water, it's the particles in it are moving and bouncing off each other faster. When it's cold, ice cold, they've stopped moving entirely. You've drained all the energy from them. The more heat you apply, the more energy you apply. So basically, when we have something called diffusion, this is when you move particles from an area of high concentration to an area of lower concentration. If I take food dye and I put a couple drops in a glass of water, right, it goes from a really, really dark, deep blue in the food dye to maybe a light blue in the glass. That's diffusion happening. Does anyone here drink tea? Okay. Yeah, when you put a tea bag in, you can see it happening live, right? Like right when you put it in, that's diffusion. You do not need additional energy for this to happen. It's really what it is, right? Again, the example here is what happens if I take some red water, some blue water, and then I put something in between it. If I take out that barrier, what's going to happen? They're going to eventually make purple. But at first, you only get the purple in the middle, right? It's still red and it's still blue. This diffusion takes time. If we were to look at this on a microscopic level, this is what you'd see down here. You would see the particles, the blue and the reds, slowly and slowly interacting with each other and then balancing each other out. So all you really need to know here is you just need to know this. This is the same thing that was over here. You just need to know what diffusion is. This is the big one there. Hold on a second. So again, a lot of people like thinking about diffusion as they think about examples. Red and blue water, what happens with that? That's diffusion. Some people like thinking about, this is the same thing. I just moved it up here a little bit smaller. If you can't see that, let me know. Again, these are all just examples. You don't have to write all these down. I would recommend, think about an example that's really concrete to you. Some people like thinking about the aromas in the air. You guys see we have these air fresheners here, right? If I take it just out of the box, I put it there, I'm not going to smell it from across the room. But if I let it sit there for for a day, I come back in the next day, I'll smell it from over there. Same thing like sugar and water has to slowly diffuse. Usually we stir it to agitate it, right? You're purposely diffusing it faster. You mix hot and cold water, what's going to eventually happen is it warms up. If you guys have ever had a bath, the water gets cold, you turn it hot on one side, the side where the water's coming from, a lot hotter than the stuff by your head or your feet or wherever or orientation you go with tubs. So it's just important to know what is diffusion. So eventually, again, you don't really need to know this. Like this is just very scientific writing of it. You just need to know that if I mix red, if I mix blue, I will eventually get entirely purple, right? That's what this is saying. This dynamic equilibrium is the purple. So you could summarize it in 2 words if you really want it. Dynamic equilibrium equals purple. That's when those concentrations become uniform. So that's just really the only big part. So what does it have to do with potassium and salt and sodium? Well, that's where we're going to kind of lead up to here. Okay. Okay, so what is about this plasma membrane? We know that every cell has a plasma membrane. We know what it's for. It takes in and chooses what it wants to have. It has selective permeability. It selects what it wants to take in, selects what it wants to give out. So we know that water can diffuse across it. So one of the things we do though is we use something called facilitated diffusion. So if you guys remember, this is a little bit of an older picture here from earlier in the week, but this is our cell membrane here, right? And we go through these proteins. That's the highway from one side to the other. Think of it like a bridge. So those facilitated diffusion uses these. We say diffusion, it uses those. So these are passive transports. They don't require additional energy. Something like that, a passive transport. Again, think about if you put red and blue into two different sides of a tub, they will eventually turn purple. Again, this is the red and the blue. Or like spraying Febreze in the air, all that stuff. Okay, so let me just I'll play this really quickly. Share. I just want to make sure it's sharing my sound too. Your sound. Optimize for video clip. Okay. Just really quick tutorial on diffusion. It's a minute.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Oops. So again, come on. So again, when we're looking at passive diffusion, it doesn't require energy. But when we look at facilitated diffusion, it's a little bit different. So again, this is just those same pictures there. We have the outside, we have the membrane, we have the inside of the cell. Water can just go in and out as it pleases. Just like your hand goes in sink, ends up all wrinkly, like you didn't really expend energy doing that. It's just trying to balance it out. But what if we're trying to bring something in? What if a cell is trying to eat something? How does it pick that? I want to eat that. It uses these proteins, right? It wants to take a sugar molecule and take it in. So it's going to grab it. It's going to launch it in. and it's done purposefully. So facilitated diffusion takes energy. It is a concentrated effort to do it. So, so next we're just going here quickly into osmosis. So this is when we're looking at, again, diffusion of water. Selective permeable membrane, that's your membrane. So it's going to move across until both concentrations of pollutions are the same on both sides. This is why if you put your hand in dishy soap water, it can eventually end up wrinkly. Because the water's trying to balance itself out. Again, if you're looking at this experiment, what's actually happening here right now, before, what we've done is we've put the same amount of water in between. We've put a membrane in between it, or water can flow through. Naturally, if you think about gravity, they should be the same level, right? But what happened is one of these sides is way more sweet. They put way more sugar in it. And so what ended up happening is a lot of this water moved to try and balance that out. It wants to get the same amount of sugar for water. One way to think about it is this right side here is really, really, really sweet. This left side is not as sweet. So it's going to balance it out, and now both sides of this are going to taste the exact same. So osmosis is the diffusion of water. And that is typically a passive form of diffusion. It is typically passive. Okay. Again, these are just some examples here. If you're really thirsty after eating salty food, it's probably trying to regulate itself. You soak your fingers, they'll get pruning. They'll get pruning. If you ever put raisins in water, they're gonna soak up a lot of that water. It's trying to balance it out. Plant roots sucking in water, that's how they do that. You put in sugars on strawberry, does anyone know what happens? The strawberries release a lot of their moisture. That's what ends up happening. So you make like a natural syrup that way, for example. So those are just some examples here. So now we're going to talk about isotonic and hypotonic and hypertonic. So a couple different things. So isotonic is when the water is the same. So the water is the same. So isotonic, same water concentration. It moves in, but it moves out. If you're thinking about traffic, you have the same number of vehicles going in a parking lot and out of a parking lot at the same time. Yeah, isotonic is where your water's the same. I recommend, try and keep it simple like that. It's easier to remember, right? Okay, so. Oops, the writing down, okay. Okay, so isotonic is when water's the same. Hypotonic is when you have a lower concentration on the outside. So water's trying to move in. This is hypotonic is when you have a raisin. Think about what happens to a raisin in a thing of water. It is going to suck up a lot of that water. So water moves into the cell. And what usually happens is the cells can burst if they're put into a hypotonic solution. This is how a lot of antibacterial things work. So they'll do something to rupture the cell wall. Again, I always recommend trying to think about like a real world example. You think about what happens with a raisin in water, you're gonna remember what happens with this. You don't need to fully have all the scientific words to really understand what's happening with it. So we usually think of examples. So again, isotonic, the water is the same. Hypotonic, the water moves into the cell because it has a higher concentration. And hypertonic is when the outside has a higher concentration. So water moves out of the cell and decreases the pressure. This is your hands in soapy water. So this is your hands in water. They shrivel up, they shrink. This is what actually wilts most plants. Okay. Everyone done? Okay, so yeah, those are the three, right? Isotonic, the water's the same. Hypotonic, it's like a raisin, water goes in. Hypertonic, It's like your hands in water, water exits it. So when you have to move from an area of lower concentration to an area of higher concentration, and without looking at your notes, which one of those three is that again? Moving from lower to higher. That would be hyper. So this is like with your hands again, from lower concentration to higher concentration. You need active transport for that. This now requires energy to happen. Right? If you try and pump water uphill, if you want water to go uphill, you need to pump it. It's not going to flow there naturally. So it needs that energy. So we are almost done. We got 3 more. So again, this active transport is, like I said, it's active, right? It requires energy. It's doing something. So how does it work? Well, like I said, this is where those electrolyte ingredients, sodium and potassium, come into play. So the pumps are sodium and potassium. Your pumps are electrolytes. So this is in your plasma membranes, and basically this tries to maintain the sodium and potassium inside and outside of the cell. So there's kind of the process here, but basically what ends up happening, these are these carrier proteins, right, on the cell membrane. So these are going to grab a hold of the salt and the protein, and then it's going to eventually just launch it up. So it uses sodium to take it, and it uses potassium to exit it. Okay, so again, so these pumps, sometimes you need to also have a large molecule move against something. So if you take a really, really small downhill thing, on your bike, you might slide down it. But if you have a really heavy thing, it might be so heavy it just stops. You have a semi on there, it's just going to stop, even if it's going downhill sometimes. It's just too heavy. So sometimes you need energy to do it. So if you have a large molecule and it needs to move against a concentration gradient into the cell, we use that coupled transport. Basically, those sugars pair with the sodium and the potassium, and they enter the body. So this lets the sugars enter through facilitated diffusion. So it just kind of like hitches a ride. There's a train going on, and it just hitches a ride. It's a hitchhiker. This isn't that important. I've never seen like a question asked about this stuff ever before. So, fingers crossed. Okay, we have one more after this. Okay, and so last one, we have endocytosis. Again, this is looking at large particles. So sometimes what your body does, if you've ever seen like someone rejecting a bullet in a movie or something or in the news, if someone gets shot or a piece of hair gets like left in their body or like something enters their body, a really deep splinter they couldn't get out. Eventually, the body will reject it. The body will push it out itself. The cells form around the object and slowly push it. It takes a couple of years, but it happens. Your cells do something similar, it's just a little bit faster. Basically, endocytosis is when a cell surrounds an object. It takes it in and then it surrounds it to accept it. So like they would basically form a wall. It opens it up and then it closes itself and it slowly gets brought into the cell. The other one is exocytosis. This is how it spits it out. So endo is going in, exo is going out. And the cytosis part is just forming a wall around it. It's forming another plasma membrane around the objects. Okay, so that's it. That's it for section 7.4. We're done with that part of the cells. We're done with chapter 7, all the fun stuff like that. So what we're just gonna do now, we got a little bit of time left. Yeah. So you'll get a chance to look at one of those cells. I've actually assigned it here. I'll give you guys a second to load it up before I go in and look what it's supposed to look like on your end. Again, this is not for marks or anything, but it's a good way to be able to test yourself if you remember it. So what I want you guys to do here is I want you to go into the animal cell and I just want you to like look at it for a minute, try and think like what each of those organs are. Which one's the ERs, which one's the nucleus, so on and so forth. Try and guess which one's what, and then you can just hover over it and look. So I'll give you a second to do that. Paige, I sent you your stuff there in the chat. They had to do some stuff, so that's why your password's a little bit different. So I'll give you a minute to get there, and I'll show you what that's supposed to look like on your end. Okay, so it's everyone on that website at least, we able to get that? If not, let me know. But eventually you should get something that looks like this, right? And so once you log into it, now for you guys, you should have access to all your books. They checked your account as well page. You should have access to it now. And then once you do that on your, now this is where it looks different, but on your guys's bottom right, you're going to see assignments. I'm going to want you guys to click that. I'm going to want you guys to click the assignments part of it. It's not an actual assignment. I've just assigned it so it's easier to find. And then you're going to click on the human cell wall, which should, what's going on? Here we go. Which should be this. Once it loads. And again, all I want you guys to do is you can zoom in, you can zoom out, you can use your left and right click to do stuff. The left click moves stuff. The right click changes your angle, and you can zoom in and out. So all I want you guys to go in here and do is there's a whole bunch of things here, right? If you hover over it, it's going to tell you what it is. I want you to go through it. I just want you to guess, like, you know, like, I think this one, you know, this, what's this big blue thing here? That's the nucleus, right? So like, oh yeah, that's not the nucleus. Oh yes, it is. So I just want you to do that. Like for example, what's this like green thing right here? Oh, maybe you don't remember. What's this big red thing here? What's this green stuff? So the whole point of this is just trying to be able to visually identify some of this information, and that just takes some practice. Is really what it is. So that's what this is. So you guys got about like 5 or 6 minutes to be able to do that. And if you couldn't access it, let me know. I'll come help you out. We're going to be doing this with a couple things throughout the course.